So, chapter 4. Now, chapter 4, we're primarily now talking about intracellular stuff, biochemistry, right? So, the first thing we have to understand is what is DNA, right? And what is RNA? How does DNA relate to our body? How does it relate to chromosomes? How does it relate to genes? All this stuff, right? So, when we look at this, DNA, right? We're not going to talk a little about Mendelian genetics, but since this is anatomy, we're not going to do like too many Punnett squares. Who remember doing Punnett squares? I know you guys all love Punnett squares, right? It's actually pretty interesting, right? It's just, it gives you the thing about what Punnett squares is it gives people the ability to see what the probabilities are in certain type of diseases. The problem is not a lot of these diseases use, Mendelian genetics where it is autosomal recessive, autosomal dominant, X-linked recessive, right? They're usually multifactorial. So you might have the ability to inherit part of that disease from one of the parent. You don't exhibit it until something else happens, right? So most diseases, especially things that we see very commonly, hypertension, right? Multifactorial. So is diabetes, right? Now, even type 1 and type 2 diabetes are multifactorial. All right. DNA, right here. Structure. All right. DNA is deoxyribonucleic acid, long thread-like molecule with uniform diameter, but varied length. So not all, remember, we have 23 different chromosomes. We have two pairs of it. So we have one chromosome number one from your mom. It's going to code for all the proteins that are found in chromosome number one. You have another chromosome number one from your dad. It's going to code for similar proteins, but it might vary on the amount you produce of that protein. It might vary on the color, right, you might have of that protein. So what we're going to see is that you have two versions of the same gene found on the same chromosome, right? What you are going to express might be different, right? Depending on which one is active. So this is where we see the variations in kids. Some kids from the same mom and dad one child might have little blonde hair, the other child might have a little bit darker hair, right? That's, appropriate sometimes, right? That's not even talking about like some recessive traits that can freak people out, right? So yeah, when we're talking about chromosomes, we have 46 chromosomes, 46 double strands of DNA, right? That's what DNA and chromosomes are, right? We have 23 of them that are distinct. Meaning what? When we look at chromosome 1 and chromosome number 2, right? Chromosome 1 and 2, they're going to code for completely different proteins, right? So if I look at a specific part of chromosome 1 and compare it to that same part in chromosome 2, they're going to code for completely different things because they're different chromosomes, right? Some chromosomes are much smaller, right? We're going to see the male chromosome, the Y chromosome, much shorter, right, than the female the female X chromosome, right? So we have some kind of things that look very similar. The chromosomes 1 to 22, they look very similar. Then we have this sex chromosomes that look very different to each other, right? Average human DNA molecule is 2 inches long. 2 inches long, right? That's pretty long. That's something we can see. That's not going to fit in our nucleus, right? Unless we roll it up. And right, DNA and other nucleic acids are polymers of nucleotide. Remember what we mentioned? What a polymer is, right? A polymer is just right, identical units bonded together, right? So a nucleotide consists of a sugar, phosphate, and a base. And this is what it looks like, a nucleotide. The phosphate group is out here attached to the sugar. In DNA, the sugar is deoxyribose, right? In RNA, right, all we have is a little oxygen sticking out right here, and now we have ribose, right? The bases, right, for DNA, the bases are going to be adenine, thymine, then guanine, and cytosine, right here. Adenine, guanine. These are what we call purines. They're big. They're double ring structures. Then you have the smaller pyrimidines on the other side. All right, guanine and cytosine. right? Adenine with thymine. Now, uracil is found in only RNA. In RNA, we remove thymine and we replace it with uracil. Now, you're going to ask me, right? Why do we have something called, right, complementary base pair? How come we see adenine on one side of the DNA strand? On the other side, you should see a thymine. Why? Right? The reason why is simple, right? Is to maximize the number of hydrogen bonds. By maximizing the number of hydrogen bonds, you keep the double strand of DNA intact, right? Right. What else do we see? Right. A single nucleotide, we kind of talk about the sugar, phosphate, and then these rings, these right, purines and pyrimidines. DNA has the ATGC. RNA has no thymine, but uracil instead. All right. They look very similar. Uracil and thymine. All right. It looks like they kind of inverted. That's all. All right. The double helical shape of DNA resembles a spiral staircase. Anybody's been to Discovery World, right? When you go into Discovery World, right, you enter and there's all those wooden kind of like, mechanical things right there, right? And then you walk through it after you play with all the little different mechanisms. It's fun, right? And then you go, and then as you walk up to the second floor, there's a double staircase. You guys know that's supposed to represent DNA, right? It's the double helical structure of the staircase right there. If you look inside, you see some kind of what looks like art that kind of moves back and forth. Those are the bases in DNA. So if you go to Discovery World with your kids, right, next time you go, take a look at that. It's kind of cool how they did that, right? But they are trying to impart this kind of structural art with our DNA structure, right? Double helical ship of DNA resembles a spiral staircase. We have a side piece is a backbone of phosphate groups with deoxyribose. We call this the sugar phosphate backbone. So the main megastructure, the ribbons found in our double helical strands, right? That is due to our sugar phosphate backbone. Probably easier to see it, right? So here are the ribbons right here, right? In DNA. Kind of wrap itself around each other, right? This double helical structure of DNA, right? What makes up the ribbons is the sugar, phosphate, sugar, phosphate, sugar, phosphate. This is why we call it the sugar phosphate backbone, right? Because the backbone of it, the main megastructure of DNA's double strand, is due to the fact that we have ribose, a deoxyribose, sugar, and a phosphate group. And then we attach another sugar and another phosphate group, right? Same thing on this side, right? Now, the bases face each other right here, right? So for every adenine, you have a thymine. All right. For every guanine, you have a cytosine. Now, why does this make sense? Right. Well, we do not want adenine with guanine. Right. If we have adenine and guanine, that means in certain areas you have one large base with two rings, and next to it another large base with two rings. So that area is going to be wider, right? So then you can fit both of these large bases sticking inside. All right. What's worse is that if these two are found facing each other, that means these two are facing each other. So in some areas, you'll have widened regions and then narrowed regions. Widened regions, then narrowed regions, right? There's no uniformity at this point, right? So what do we do? For every larger structure, we need a smaller one next to it. So adenine can either be with thymine or cytosine, right? Large one with a much smaller one. So how come adenines with thymine instead of adenines with cytosine? Maximizing the hydrogen bondings. We put adenine and thymine together, we have two hydrogen bonds. More importantly, when we put guanine and cytosine together, we have three hydrogen bonds, right? Without these hydrogen bonds, the double strand of DNA falls apart, right? The only reason why they move around each other, right, and form this double helical structure is because of the hydrogen bonds, right? So we have this really tightly rolled double strand of DNA, right? In order for us to replicate it, to make copies of it, so that we can double the DNA and then split the cell in half, right? To make two cells from one, right? Mitosis. Well, in order to do that, we need to unroll it a little bit, right? How do we do this? We break apart the hydrogen bond, right? So by breaking apart the hydrogen bond, we can unroll the double-stranded DNA. So what happens if we break apart all the hydrogen bonds, right? You're not going to have a DNA left, right? That's a bad thing, right? So let's talk about a couple things, right? And then, so this adenine, cytosine, again, adenine looks like this. You're not going to need to know what it looks like, right? You just need to know what goes together, right? Adenine with thymine, guanine with cytosine, right? Now that we know this, then a couple things happen, right? This right here. Law of complementary base pairing, right? Means that by knowing the number of one of the bases in a given region, not only do you know the number of the other complementary base, but you should know all of them. What do I mean? Right? Here, if I have a region of DNA that has 15% of that region, so a very long strand of DNA right here, and that piece of DNA comes out, and this piece of DNA, right, when we measure, right, it has 15% adenine. How much cytosine is it going to have? It's not 15%, right? How do we get this? Right? Simple, right? If we have 15% of this region is adenine, what other thing matches adenine? Thymine. So we should have 15% thymine. You add it together, that's 30%, right? 30%, right? Cytosine equals the number of guanine, right? We only have those four bases, right? Out of the four bases, we have 100%, right? We only have four. 1, 2, 3, 4, right? When we have all four, we have 100%, right? That's all it can be, right? So what happens? The remaining two make up the remaining 70%, right? Exactly, right? What happens? You divide it by 2, 35% is going to be cytosine, 35% is going to be guanine. All right. So by knowing the number and the presence of one of the bases, you know the numbers of all of them. Right? Same thing if I can give you another example, right? If I have 10% guanine, 10% of it is guanine, right? How much adenine am I going to have? Right? 40%, right? Why? If I have 10% guanine, that means I have 10% cytosine. That's going to make up 20%. These two then need to make up the remaining 80%. And they're found in equal numbers. What's 80 divided by 2? 40. Right? So by knowing one, you should know all of them. Right? So we'll finish this up right here about chromosome and chromatins. And then we'll stop before we kind of go into too much detail about protein synthesis. So let's just finish this up. All right. Again, what is a gene? Right? A gene, information containing segment of DNA that codes for the production of RNA that plays a role in synthesizing one or more proteins. All right. Now they go a little bit more detail here, but the basic fact is the simplest way to understand it is to know that each gene is going to code for proteins. All right. It's a sequence of your DNA molecules, the ATGC, right? That sequence is going to code for certain amino acids, right? As you add those amino acids together, we make a protein. Amino acid sequence of a protein is therefore determined by the nucleotide sequence in the DNA gene. right? We call that sequence, amino acid sequence, the primary structure. So what the DNA gene tells us, right, is that it's going to code that all those ATGCs are going to code for which amino acids goes where, which is our primary structure of the protein, right? Again, we have about 20,000 different genes. The weird thing is this, right? Remember, we thought we are gonna have hundreds of thousands of different genes, right? We have a tenth of it. Well, it turns out that only 2% of our DNA is actually functional genes, right? It codes for something. The remaining 98% is non-coding, but that doesn't mean it's unimportant, right? What it does is it regulates the gene activity when we make that protein, when we activate the gene, when we deactivate the gene, how much, right, do we want to produce from that gene, right? All of that are regulatory regions. So 98%, even though it's not coding, does not mean it's not important. Because again, you can make insulin at the wrong time, and that's a bad thing, right? You need to know when to make it, you need to know when not to make it, and you need to know how much to make. That's all part of the non-coding regions of DNA, right? Chromatin and chromosomes, right? Chromatin, fine filamentous DNA material with proteins, right? Occurs as 46 chromosomes in most cell. Now, when we see in a second here, right, we call each one of these right here, right? Whenever you see a double arm structure like this, right, we have double the DNA. A normal chromosome only has one arm. When you see a second arm next to it, that means that we've undergone DNA replication in the cell, right? And DNA replication means you've replicated. You've, kind of, right, doubled it now. Instead of one chromosome, you have a second one next to it. The weird thing is this, right? When they're attached to each other in the middle, by this area we call the centromere, they count as one chromosome, right? Even though there's two, enough genetic material for two. But since they're attached, it only counts as one. As soon as we detach it, even a 1/10 of a nanometer, just a little bit of space is seen, they count as separate chromosomes now. Weird, right? All right, so when you see this, they'll talk about how this is one chromosome. It is, but it's got the genetic material for two. It's only called one chromosome because they're attached to each other. As soon as they slightly detach, they're separate chromosomes, right? So what do? We can't say this is just one chromosome, right? And expect you to understand, this is just one chromosome, but it's got two arms, right? Well, we call each arm a sister chromatid, right? So a chromatid is an arm, and you should have a sister chromatid next to it, right? When we have two, right, when we have sister chromatids laying side by side, with double the DNA, we should have genetic material for two chromosomes, but they're attached to each other now, right? When they detach, they will count the separate chromosomes, right? So, yeah, just be careful when you're reading this because they'll talk about how you double the DNA through DNA replication, but yet you don't double the number of chromosomes, right? Why? It's because they stay attached to each other by a central protein called centromere. When we break apart that central protein, right, and then one cell gets one copy and the other cell gets the other copy, they count as separate chromosomes. So you have genetic material for two, but they only count as one because they're attached. All right? All right. What else? Those are proteins that allows us to roll the 2-inch DNA molecules into small enough, compact, condensed genetic material that we can hold in a nucleus, right? DNA molecules wind around a cluster like thread around a spool. Think about what happens when you see a piece of thread and you start rolling it up into a ball. That's exactly what happens here, right? You take this long thread like DNA molecule and rolling around a protein. So then it's smaller, it's more condensed, and now we can package it in the nucleus, right? Nucleosome consists of core particles, histone clusters with DNA, linker DNA. A lot of this is a little bit beyond the scope, so I wouldn't ask anything about this, right? Just know that in terms of chromatin and chromosomes, right, and DNA, how they're related, right? Chromatin is really condensed. Usually we see it a sister chromatid with it because it's a sign that we've undergone DNA replication. When we split the sister chromatid, they become separate chromosomes again, right? So chromatid, chromatin is what we call it when it's in the process of either being replicated or it's in the process of cell division, right? Once we're done with cell division, we call it chromosome again, right? In a non-dividing cell, the chromatin is not static. They change moment to moment according to genetic activity. All right, what do we mean by this? All right, we package the chromosome so tight to fit into our nucleus that remember, it has a function as well, the DNA, the chromosome, right? The function is to code for a protein. So when we need the protein, what do we have to do? You have to unroll it a little bit, right? This is why what it means by it's not static, right? At any one time, at any one cell, different parts of different chromosomes can be unrolled so then we can read it and make specific proteins from them, right? Again, the weird thing is this, every single cell in our body has the same genetic material. So how come only the cells in your pancreas, the beta islet cells, produces insulin, right? How come the cells in your cheeks only produce saliva, right? Well, it's because in certain areas, right, the chromatin in those areas are not quite active, right? And it has to do with location, right? Certain genes get turned off, certain genes on based on location here. Right, almost done. I know you guys are about dying on me now. All right. Chromatin and chromosomes. When prepared to divide, cell makes a copy of all nuclear DNA. Right now they emphasize nuclear DNA because there's DNA somewhere else, right? Mitochondria. Right. So before we divide, the mitochondria, right, move to different positions inside the cell. So then each cell will have some mitochondria because it's not coded for in the nucleus. Right. So we're going to double the chromosomes. We're going to double the amount of, right, cytoplasm. We're going to double the amount of organelles in the cell. And then we're going to split everything in half, right? Each chromosome consists of two parallel filaments of identical DNA we call sister chromatids. So here's one sister chromatid. Here's another one, right? Again, since they're attached, we say it's just one chromosome, though. I know, right? Isn't it crazy how we use these wording? All right, don't even let me talk about, right, what happens with our ploidy number, 23N, all right, 2N. It becomes very confusing, right? So we won't talk about it much, right? Again, chromatids are joined at the, all right, constricted middle part called a centromere region, right? Proteins connected together. And then you have proteins on each side of it right here that will then eventually allow us to pull it apart, right? As we pull it apart, because remember, this is a copy of this, right? So as we pull it apart, each cell should have one copy of one chromosome from each parent. Perfect. One chromosome number 23 from mom, one from dad. One chromosome number one from mom, one from dad, right? Perfect, right? So we split everything in half. So then this copy goes to one cell, the other copy goes to another cell. One copy of one chromosome from each parent, right? We'll talk more about that next week. All right, I'll stop right here. Any questions?